Hello everyone, I welcome you all to my channel. Today we will discuss whether mutation is harmful or useful. In my last video we have discussed the meaning, causes and types of mutation. Let's begin. Mutation can be termed as harmful and useful depending upon the result in a particular situation. First we will discuss the examples or the cases where mutation can be termed as harmful. Let's begin. Suppose there is a chromosome number 5, we are taking as example. As you all know, every chromosome has an exact copy which is called as homologous pair. So this is a pair of homologous chromosome. Let's suppose there is a gene on this chromosome number 5. This is the homologous pair. So exact gene will be present in that chromosome also. Let's suppose this gene, which can be termed as P, let's suppose this is termed as gene P. This gene P forms a protein which acts as an enzyme named as phenylalanine hydroxylase. When this gene is normal, it forms a functional phenylalanine hydroxylase which converts phenylalanine into tyrosine. These are the types of amino acids. When this is functional, phenylalanine can be converted into tyrosine. But if a mutation takes place, let's suppose in chromosome number 5 in one base pair, let's suppose in gene P, this is the sequence, let's suppose, of gene P on chromosome number 5, and this T has mutated to G, let's suppose. The same type of mutation if takes place in the homologous chromosome also, that is both the genes present in both the chromosomes, if undergo mutation, then there will be no functional copy of phenylalanine hydroxylase, as these both are now changed sequences. So the protein which will be produced from these genes will be abnormal. That is, they will not be same as that the previous case. The enzyme which will be produced from these abnormal proteins will not function as phenylalanine hydroxylase, as a result of which phenylalanine will not be converted into tyrosine, and this phenylalanine will accumulate in our cells and they can convert themselves into phenylpyruvic acid or other derivatives which are very much harmful for our body. This accumulated form of phenylalanine leads to various disorders like mental disorder, and this condition is called as phenylketonuria. So phenylketonuria, I have just taken one example. We will see many such examples in our upcoming videos when we will discuss types of mutation. So in this case, mutation can be termed as harmful. Other such examples of harmful effects of mutation are Down syndrome, Turner syndrome, then Cry-du-Chat syndrome. We will discuss each and everything in detail. The other case where mutation can be termed as harmful is cancer. All the cancers that are generated in a living organism is due to mutation. How? Every cell has few genes which control the number of cell divisions. That is, suppose this is a parental cell, it will divide to form two daughter cells. So this is one event of cell division. This event of cell division is controlled by many genes. Suppose there is a mutation which has taken place in one such gene which controls the number of cell divisions. In that case, the number of cell divisions controlled will be failed, as a result of which this cell will undergo uncontrolled cell division. That is, instead of dividing into two daughter cells, these daughter cells will keep on dividing and will generate faulty cells in every generation. As this cell was faulty due to mutation, and this gave rise to these two daughter cells, and instead of remaining at this place, the cell division will continue generation after generation, and we will get a bunch of such diseased cells. This was due to mutation in such gene which controlled the number of cell division. As you all know, cancer is uncontrolled cell division. The other case when mutation can lead to cancer is mutation in DNA repair gene. As you all know, during replication, some mistakes take place which are called as errors in replication. Let's suppose these are the two strands of DNA, these are the parental strands. During replication, In place of A, T will be incorporated. C, G, C, G, A, C, G, C. This is the normal replication process, and these are the daughter strands from. These are copied from these two parental strands. During this replication, let's suppose instead of this C, a T is incorporated. This is an error in replication. This error is corrected by the help of DNA repair mechanisms. DNA repair mechanism consists of some proteins which remove this T and incorporate a correct C. Let's suppose a mutation has taken place in gene that forms this protein which will correct this error. In that case, this error will be present or prevalent, or it will remain there, as a result of which the cell which will be produced because of this will be diseased, and then when that cell will undergo cell division, we will get a whole bunch of diseased cells. The cases when mutation can be termed as useful are as follows. Mutation leads to variation, and sometimes mutation can also lead to speciation. How is it possible? Let's suppose there is a sequence A, T, T, C, G. Due to mutation, we got a sequence A, C, G. These two are mutated into AA. Let's suppose this gene forms a protein that forms, let's suppose blue colored eyes. Due to this mutation, a new type of protein is formed which forms, let's suppose, brown colored eyes. Here, due to mutation, the protein which is formed is different from the original, but it is functional and it forms a different type of eye color. So in this way, mutation can lead to variation because many types of eye colors are present in population, which can be also termed as polymorphism, as different types of eye color, different types of hair color. These are the result of mutation. In all these cases, the new proteins which are formed give us a different result from that of original one, as a result of which variation is produced. What is variation? Variation is difference in DNA sequence from that of the original one. That is, new type of proteins or new types of alleles or new types of genes are produced in these cases, which give us different phenotypic results. Thus, mutation is the raw material upon which natural selection acts, and mutations which increase the survival chances and help human beings to adapt better are selected and passed on to the next generation. Now let's talk about a condition when mutation can be considered as useful. We will talk about sickle cell anemia. It is a genetic disorder in which there is a deficiency of hemoglobin in blood. It takes place due to mutation in gene that forms hemoglobin protein. As you all know, we have 23 pairs of homologous chromosomes. So let's suppose chromosome number 5 is exactly similar to chromosome number 10. So these both are homologous chromosomes. Let's suppose the gene that forms hemoglobin protein is found on chromosome number 5. It will be also present on chromosome number 10. If mutation takes place on both these chromosomes, that is in both these genes, then a faulty hemoglobin protein will be formed. As you all know, an RBC molecule contains around 300 million hemoglobin proteins. Due to these faulty hemoglobin proteins, the shape of hemoglobin will be changed and hemoglobin will take a C shape or a sickle shape. But in some cases, the mutation in gene for hemoglobin takes place in one location only. That is, let's suppose this mutation took place in gene on chromosome number 5 and not in gene on chromosome number 10, because of which half of the hemoglobin proteins produced in body will be normal and other half will be abnormal. This condition is called as sickle cell trait. Sickle cell trait is a condition where symptoms similar to sickle cell anemia are not seen. That is, mostly such people where only one copy of gene is mutated have normal life, but they can transfer or pass on the mutated gene to next generations. Sickle cell trait is advantageous for survival of individuals in places where malarial infections are prevalent, especially in African subcontinent. the people who have only one copy of mutated gene have survival advantage, because once they are affected by malarial parasite, because of sickle cell trait, that is one copy of gene is mutated, that parasite is not able to take over that individual, that is it is not able to affect that individual as compared to normal people who have both the copies of the gene as normal. Even though mutation is common, inherited diseases or genetic diseases are relatively rare. This is because inherited diseases are often recessive, which means a person must have two copies of the mutated gene in order to get the disease. This is a gift from nature which helps in survival of human beings. A single mutation can have a large effect, but in many cases, evolutionary change is based on the accumulation of many mutations with small effects. Mutation effects can be beneficial, harmful, or neutral depending on location. Most harmful mutations or non-neutral mutations are deleterious. That means the individuals who have harmful mutations in them mostly die before the reproductive age, or even if they are able to survive till the reproductive age, they are not able to produce children. Thus, mutation can lead to variation, speciation, and evolution. Speciation is the formation of new species. With this, we come to the end of this video. In the next video, we will talk about frame shift mutation and various other types of mutation in upcoming videos. Thank you for watching.